Genomics: Observing Evolution. Introduction to terms. Genomics is the study of genomes. A genome is the complete set of genetic information that an organism needs to function. It is a set of instructions that influences, along with an organism's environment, how every cell in that organism's body will grow and develop. And that is true for all living things on the planet. Genomes are comprised of DNA. DNA, short for deoxyribonucleic acid, is a long molecule made up of 4 repeating units called cytosine, guanine, adenine, and thymine. Incredibly, these same 4 molecules, called nucleotides, encode the genetic information needed for every living thing on Earth. In a DNA molecule, the 4 repeating units create 2 chains, with adenine always pairing to thymine and cytosine to guanine. These sets of bonded molecules are what we call a base pair. The chains coil around each other in the form of a double helix, which is wound up tightly and packaged into chromosomes. Nearly every cell in the body carries a full set of chromosomes in its nucleus, and therefore a full set of the organism's DNA. This complete set of DNA is the organism's genome. Each chromosome has specific sets of DNA arranged as genes. Genes are the basic units of heredity. They are, biologically, what makes you a unique member of the human species. The human genome consists of approximately 3 billion base pairs. If you were to stretch out the base pairs from a single cell in your body, they would span over 6 feet. If you were to stretch out all of the base pairs from every cell in your body, they would span the entire diameter of the solar system twice. In a human cell, the 3 billion base pairs are distributed across 23 pairs of chromosomes, but the number of base pairs that make up a genome varies across organisms. For example, a banana has about 500 million base pairs across 11 pairs of chromosomes, while a fruit fly has over 139 million base pairs distributed across 4 pairs of chromosomes. But while every organism within a species shares the same amount of DNA, the sequence of their DNA is never exactly identical. Individual organisms of the same species will share most of the same DNA sequences. Any 2 humans share about 99.9%. However, they will have sequences that are different from one another. These distinct genetic sequences are what make every organism unique, even within their own species. The difference in genetic characteristics within a species is what we call genetic diversity. Think of a genome as a cookbook. A cookbook contains recipes needed to make a meal. Each recipe lists the necessary ingredients as well as directions for preparing and cooking the dish. In the same way, a human genome contains genes that include the ingredients and instructions needed to make a person: muscles, bones, organs, nerves, and the rest of the tissues that make up a human being. In cooking, some changes to recipes have no effect on the end product. Some are disastrous, and others may actually improve the dish. The same is true of genes. Genetic sequences change or mutate for a variety of reasons, like exposure to environmental factors and fluke accidents during the replication process. Mutations can have neutral, negative, or positive effects on the individual organism, and ultimately the species. Evolution is the process by which genetic information changes within a population as it gets passed from one generation to the next. But evolution isn't always something we can see with the naked eye. Genomics allows us to see changes in DNA sequences at a molecular level, giving us a new and powerful tool for observing evolution.